All right, I think we can get started on the first couple of slides. I'm Rachel Hostetler, and I'll be moderating this webinar today featuring Matthew Schmitz and Nelson Johansson. And today they will be talking about a new project of creating a cross-species spinal cord taxonomy. And just a few general notes and reminders for this webinar. Again, this is being recorded and it will be going up on the YouTube channel afterwards. Our YouTube channel is at Allen Institute. And if you would like to ask any questions throughout the webinar, we will be answering those questions at the end. We'll have about 10 to 15 minutes. And you can use the Q&A function in Zoom to ask those questions throughout the webinar. All right, now I'm going to hand it off to Matthew and Nelson.
Great. Thanks, Rachel. So today we will be talking about, oh, sorry. Okay. It is a little bit laggy. We'll be talking about some work which you can read about on bioRxiv. And this is sort of a little bit of the results and a little bit of how the sausage was made. And so to sort of give you a picture of how these taxonomies get built and then what you can do with them. And so today we'll be talking about the spinal cord, which is this wonderful extended motor, which is referred to as efferent and sensory afferent routing system for the brain, which includes a bunch of interneuron circuits, which do sensory integration and sort of make a compressed signal that gets sent from your body to your brain, and also enables things like your reflexes. And so the flow of information is that information comes in through the dorsal part of the spinal cord, in the dorsal horn, which we'll talk about today. And then information goes out through motor neurons in the ventral horn of the spinal cord. And right, so the spinal cord is divided into segments, where each segment, the fibers that come from it are bundled into a nerve, which goes out through a corresponding vertebrate. Interestingly, these segments don't actually line up with your vertebrae, which is it's kind of fun. So they send these long nerves that end up lining up, even though it kind of floats freely in the middle of your vertebral column. And so the spinal cord is a very special part of the brain. It's one of a handful of regions that are conserved with essentially the same function across all of vertebrates. And you can see that for a very large evolutionary distance, the spinal cord has sort of maintained this structure of sort of a dorsal horn and a ventral horn. And so it's widely thought to be one of the most conserved regions of the brain. On the other hand, more recent molecular work has shown that even though, say, the zebrafish and the mouse appear to have very similar constituent cell types, that these sort of circuits that get set up, they're a very variable complexity. Like a mouse has many more types of interneurons and performs more complex computation in their spinal cord than a bony fish does. So yeah, so our question is sort of taking a look at within mammals, consolidating the atlases that exist, as well as some data that we're generating, to sort of answer this question of what's conserved and what's not. Because there's this challenging question of cell types that can align fully or partially when you look across species, and then trying to give them names that will be analogous and will be recognizable, whether you're looking at a mouse or a human. And so this is something that a cross-species single-cell genomics atlas can help with, because you can get this very in-depth profile of cells that you can use to translate your findings from model animals to humans. And then also, another important part of this is spatial transcriptomics, which allows us to link our transcriptomic types to where they're found in the spinal cord, which really opens up looking at sort of the vast literature of anatomical work that's been done, and figuring out who is where, and that gives you an idea of what they're doing in terms of neurons and glia. Um, right, and then we're also going to talk about some epigenetic profile, which will reveal some transcription factor codes, um, and how this can enable cell type targeting with adeno-associated viruses. Um, all right, so in our atlas, we generated 10x multiome data from a number of species. In human, we have 3 donors, and we have in this paper 21,000 nuclei. In macaque, we have 3 male and 3 female with about 100,000 nuclei. We also tried to enrich for motor neurons using FACS sorting, and then we also performed MERFISH to get spatial transcriptomics. Um, and then we also aligned to mouse, which is sort of a parallel study, which will be posted on bioRxiv soon. Um, Thanks to Cindy and Yuan and to Jen here at the Allen Institute. Um, and so these are the sort of sources of data that are all getting pulled into this data set that we're going to put together and compare how these spinal cord cell types look across species. And now I'll hand it over to Nelson to talk about the taxonomy.
Great. Thanks, Matthew. Yeah, so we did this amazing profiling, across species using multiomics, but how do we build a cross-species taxonomy? That's what I'm gonna try and give an overview of. But first, I think it's helpful to reflect on the Institute's work in developing taxonomies across brain regions. This is a great diagram that Jerry Miller put together that shows on the x-axis different taxonomies in their data publication, along with on the y how comprehensive they were. So you go from visual cortex in 2016 to a very comprehensive whole brain atlas in 2023, and then extensions of that, more recently. As you can see, we kind of learned from our previous work. These connections say essentially taxonomies are evolving units through the brain. And that was great for the brain, but spinal cord, this is our first real cell-typing effort in that region. Other folks have done amazing work outside the institute that we rely on quite a bit. But this is our first atlas of spinal cord cell types. So how did we go about creating this taxonomy across species? Um, what is little laggy, isn't it? It's not moving for me. Hmm. We might just want to share individually. This is
Yeah, do you want to try again, Nelson, or should I stop sharing this?
um 1 second. Yeah, it's just not moving for me. Okay. It works so well for you, Matthew. Can you
You,
actually,
you,
can
you can
one of
try
you move?
re-requesting the control.
Okay.
No, we should be.
There we go. Okay. Sorry about that, everyone. Technical difficulties. Okay. So what is the taxonomy and what is the process to create it? The multiomics basically gives us measurements of the transcriptome and epigenome per cell. And if you think about it, those cells as they come to us are just unorganized, unlabeled pieces of genomic information. And our goal is to organize them into a hierarchy of cell types that, like Matthew said, have names that are reflective of the field and are identifiable to folks. So at a high level, what we do is we take those single cells, we cluster them based on the transcriptomic, the RNA-seq data, into these sets of cells, and then organize those clusters into a hierarchy. We call it a taxonomy that progressively defines more granular types as you walk down from, spinal cord to the really defined dorsal and ventral horn populations. So that's the big picture. How do we get from cells to a hierarchy? Um, there are a few different routes to go. Especially in these days, it's common to use prior taxonomies. So, in my mind, you have two options. One of them is this is the first taxonomy you've seen for a region, and no one else has done work there. So you have to use a lot of manually curated, you know, going through papers and finding marker genes of cell types and really pulling this hierarchy together manually. Whereas another approach is to build upon prior work, where, prior taxonomies have done sequencing. They defined a cell type landscape, and you can start from there to then, expand based on, the more diverse profiling that you may have done in your sequencing efforts. So I think we do a bit of both of these for spinal cord, given this is the first Allen taxonomy of spinal cord types. But we build off of prior studies from, Ariel Levine's group, the Gittler lab, and others. So, how do you handle cross-species data? It's one of the first things we have to think about when looking at this single cell expression. And unfortunately, with cross species, you have what are called batch effects. So every species has a unique pattern of transcriptomics in its cells, gene expression that would separate your cells first by species and then by cell type. So our first goal is just to build a taxonomy of cell types. So we want to remove the species effect on expression, and we do that through alignment algorithms like SCVI, which is one that we use in this case, but also there are approaches like Seurat and linear methods like Lima to correct for, say, species, donor, just unwanted variation that isn't important to the process of building taxonomies. Not to say it's not important for research in general, but we wanted to find cell types that work across species, across donors. So we remove those effects first before doing any other step. And this UMAP on the right is showing the result of that alignment process. So there are cells from human, macaque, and mouse in this UMAP. It's a bit hard to see, but take my word for it that there is really good mixing across species in this space that removes that species expression effect. So once you've removed the species effect, then you can go in and do clustering. Clusters, again, are those like minimal units that we try to organize into a hierarchy. There are many ways to approach clustering. Usually you start from some highly variable genes, you do some dimensionality reduction, then cluster your cells in that space. We use an approach pioneered by Zhejian and folks that tries to account for um, differentially expressed genes in every cluster to try to avoid over-clustering. Um, this is called Scratch HiCat or the transcriptomic clustering pipeline in Python. So it's a pretty robust method that tries to find the minimal set of clusters that have identifiable features, differentially expressed genes. So once we have clusters, then the goal is, well, let's figure out how to put them into cell types. And there's many ways to go, but first you need to make sure that your clusters are all good clusters, clusters of good cells to go forward with. You know, in these technologies, RNA-seq, what have you, there always is the collection of lower quality data. And lower quality means many things. You could have cells that are not doing so great. They have high mitochondrial reads. You could have what are called doublets, which is two or more cells were profiled in a single experiment. So your expression profile is the kind of admixture of multiple cell types. Or you just have, low reads, low genes detected, and this could be a quality issue, or it could be a real property of cells. Small cells have fewer genes, fewer reads. That's a harder one to, use properly. But anyways, the goal is to take our clusters and try to trim them down to just clusters of good quality data that will help us build this hierarchy. you know, this is a pretty drawn out process with quality control. There are many, many things you can look at. Showing here another example of, the bases aligned, and like how well the alignment is doing, and how well the reads are mapping the transcriptome. Each dot here is a different sequencing library, and you can see that, poor alignment or leads to poor transcriptome alignment, which means that those cells have, probably very messy gene expression profiles. And really, this quality control process requires you to look at a lot of different aspects of data. But you can get through that. And the next step is, again, organizing clusters into a hierarchy. And I think pretty much nowadays everyone tries to use a reference taxonomy to build off of. And the way you use those, at least in part, is through annotation transfer. So Yadavidal is a study of human lumbar spinal cord from Ariel Levine's group, and Russell is a study of mouse spinal cord across multiple segments. So these provide kind of an initial landscape of cell types for us to work from instead of starting from scratch. So with tools that the Allen Institute, offers, like Map My Cells, you can take these single cell taxonomies from both these studies and map their labels onto our cells and our clusters and kind of get an initial landscape of what the cell types look like. It won't be perfect, but it gives you a good starting point. Um, and, I think Memory Cells is a great tool to use, but like more recently, AI agents have become also pretty adept at describing like clusters of cells based on gene expression by going to literature and seeing, oh, this cluster has this marker gene. It must be this cell type. So we're trying to find ways to use AI in this process too. Okay, so you get some labels through annotation transfer. And now you want to start organizing your types into your clusters into higher level types. From knowing the literature around spinal cord, it's apparent that there are multiple like high level classes of neurons: glutamatergic, GABAergic, cholinergic, and motor neurons that are a subtype of cholinergics to some extent, and of course the non-neuronal cells. So the annotation transfer might not have had this high level. So we go back and look at marker genes for these various, we call at least class level of the hierarchy. And you can use individual marker genes in some cases to understand for a given cluster which of these like neurotransmitters or populations that cluster corresponds to. And these are all genes that have been reported in, literature from previous works that typically work well. It seems to annotate these broad classes. So the first step is you take all your clusters and you figure out what is the broad category of cell type that those clusters correspond to. And then you have kind of a handle to start looking more closely at what are the subtypes of glutamatergic or GABAergic cells in the spinal cord. But it's always, we find it easiest to start with the most broad types first because they're usually the most easiest to identify, again, based on pretty clear marker genes. So the next step is to start to subdivide these class annotations into finer types. And thankfully, this is what the Yadavidal and Residol study gave us through the annotation transfer, is a finer degree of subtyping below the broad gluta, GABA, and other class level terms. So you can use the labels transferred from Yadavidal for every cluster to figure out that, okay, glutamatergic cells, they fall into both the dorsal, mid, and ventral categories. And we get that initial like landscape of cell types. But it turned out in our case that, Yadavidal had not totally describe the motor neuron landscape. So we had to extend those annotations to include both skeletal and visceral motor neuron labels, again, using marker genes from literature to distinguish those subpopulations of motor neurons of the spinal cord. So your references only get you so far, and you have to know kind of what types are missing that you're interested in uncovering. So once you have these, labels and you're pretty confident in them, our approach is to start to engage with other teams to make sure that we're naming cell types in the right way. If we've called something a dorsal glutamatergic type, those cells had better be in the dorsal horn of spinal cord. So with our spatial team and the MERFISH data, we can ask the question, are the dorsal cells that we've called like glute dorsal, are they really localized in dorsal horn? To help refine nomenclature and make sure our names really reflect the biology that they're trying to convey. Um, that's one aspect, bringing in the epigenetics, so viral tooling, anatomy to understand all the different complex parts of spinal cord where we expect to see different nuclei is really important. And then, of course, cell ontology annotations of previous knowledge in the spinal cord area, as well as close work with collaborators, is a all of these things form a tight feedback loop between the taxonomy names and the kind of organization that we're defining. Okay, so we've, worked with our team to figure out the hierarchy. We're happy with it. We found it to be very useful to ensure that when we call a cell type in human, that matches exactly with the same type in macaque and mouse, our species in this study. Jesse Gillis and team have built this maiden neighbor algorithm that allows you to kind of ask the question, uh, how related are cells across species in this case to each other for a given type? And what is the best hit? What is the best matching cell type? Across species. So this map on the left is meant to convey how well our cell types agree across the three species. So the rows and columns are a hierarchy level called group that Matthew will get into a little bit more later. But the idea is you want to see the block diagonal here of little 3 by 3 matrices that say, these cell types really are closely related across all three of the species. Um, Maybe a bit easier to see as a graph. So on the right, for example, each dot is a different species, blue human, green macaque, orange mouse. And we can look at glutamatergic, for example, and these dots are connected based on similarity of that cell type across the species. And ideally, because we have three species, all these would form cliques of three with no edges outside of the cliques. But types are very related to each other. And you can kind of see that as you see edges kind of crossing types. But for the most part, we see a pretty strong clique of three for all these types across species, implying that when we say like glute dorsal 4, that means the exact same thing in a human, in macaque, and in mouse. We also are starting to use more tools to engage with the broader community. Once we have an initial taxonomy, we want to kind of pass that by our collaborators and just the public in general. So we've been using this cell annotation platform, CAP, from the Human Cell Atlas. Essentially, it's a platform where we can put our taxonomy, our molecular data into the platform, and anyone in the world can go and give feedback. So in this panel, you have on the left the hierarchy, in the middle the molecular data, on the right, differential expression, and you can paint gene expression on the UMAP. But the cool thing is that for every cell type, you can thumbs up, thumbs down, or leave a comment about why you think that's correct, or maybe it needs to be split more, or the name is potentially wrong, given particular gene expression. So we're super excited about enabling folks to give feedback to our taxonomies and hope that this can lead to better names as well. So nomenclature is really important to get right. And we've been trying to do a better job of that in spinal cord by having a really clear nomenclature system that defines anatomical, gene expression, and neurotransmitter, which provides quite a bit of information as compared to previous labels. So the example here is GLUT D1, so glute dorsal horn type 1 from Yodavidal. We've renamed to spinal cord lamina 1 through 3, expresses the MAF gene and is glutamatergic. So really a lot of rich information in this name as we've expanded it from the prior taxonomies. So this product is available. You can go there now if you'd like and poke around and thumbs up, thumbs down, leave comments, and we'd greatly appreciate that. And hand it back to Matthew now.
All right, so after going through all of that process, oh,
I will
Do we
switch
lose
that back
the
just a moment.
please hold.
Having some more technical difficulties. Okay, we'll switch this in just a moment. We were on the dendrogram slide, correct? Of the markers.
Yep.
Perfect. We go back there.
Perfect.
Yeah, great. So this is sort of an introduction of after doing all that work, the shiny new taxonomy that we're putting out here. And so, yeah, Nelson sort of gave us an introduction to all these things, but here on the bottom, you can see our groups. And this is sort of, it's a qualitative level. It's sort of like the genus of phylogeny, is how I think of group, where it's one or more clusters, sort of this finest resolution. In the taxonomy, grouped together in sort of a qualitative way, where you can assign a marker to it that's consistent. And at least that's what we go for. You can see that some of the groups have mix in the name, and this is sort of us being honest about where we're not sure whether or not things need to be split further to be consistent groups. Yeah, but so you can see from this sort of metadata that all of our groups here are found across all of the different segments of the spinal cord, from cervical to sacral. You can see that, yeah, they're grouped from between 1 and like 12 different clusters per group. And yeah, you can also see that these range in terms of how common they are. Then also we can get markers. Yes. So you can see that these groups are marked pretty consistently by at least one sort of consistent, specific, and importantly conserved, where this is sort of a little pie dot plot where each color is a different species. And so you can see where it's a complete circle with the dark color that that's a gene that's expressed in all 3 species, specifically in that group. And so this is something that taxonomies that aren't necessarily consensus, where you're only looking at one species. If you only look at one species, you really don't know if the marker that you pick is going to end up being a good marker in other species. But having this context allows us to give these markers as a resource for people, so that you can, if you're doing a study on human disease and you have a mouse model for it, you can use the same marker to find the same cell type. And yeah, so we also have sort of a little sidecar taxonomy. It didn't really fit well into our levels, but so we have sort of the first consistent sampling in depth of primate motor neurons, because we were able to enrich for our macaque motor neurons. And so we're able to pull out a bunch of different motor neuron subtypes that are well known from electrophysiological and anatomical studies, where we have alpha motor neurons, which can be divided into slow and fast firing. We also have gamma and gamma star motor neurons, and then many different types of visceral motor neurons, where there's a great diversity of different visceral motor neurons, because they control different organs and have sort of different needs, because controlling a muscle is one thing, you just kind of contract, but each organ sort of needs specialized control. And that's where the visceral motor neurons come in. All right, so we can take our taxonomy then with the gene expression profiles, and we map it onto spatial transcriptomics data. Here are MERFISH data, where on the left you can see where we've by hand sort of tried to sort of paint on some regions of interest based on traditional rexid laminae. And then on the right, you can see colors for the cell groups, where you can see in the ventral horn, the red and the orange, these are different types of motor neurons, and in the dorsal horn, you can see many different colors of these different lamina for these neurons that are processing sensory input. And so if we look at how we've segmented it, and we see where or which lamina different cell types are found in, you see that actually these groups, they're quite spatially restricted, where pretty much all of the cell types in the spinal cord are found in only 1 or 2 lamina. And this is different from other parts of the brain, where, say, in the cerebral cortex, you'll find similar types of, say, interneurons across many cortical layers. Yeah, and then so if you look at even below the group at the cluster level, if you go dorsal to ventral, you find that the clusters are even more clearly laminar. And yeah, so this is true across the cervical, thoracic, and lumbar parts of the cord. And then the lamination gets a bit more mixed in the sacral, as you can see. All right, so something we noticed when we were sort of doing some UMAP gazing is that along the left, sort of these purple cell types, these are dorsal horn glutamatergic neurons. And we sort of qualitatively noticed that these fall sort of into a sort of progression, like a sort of gradient-like structure, at least in the UMAP space, which, yeah, kind of looks like this. And so we were wondering if in fact, this is not just a UMAP artifact. And so we used this PAGA method from Fabian Thies's lab, which uses a KNN graph to sort of ask which clusters are most connected to each other in terms of which are most similar in terms of transcriptome. And so the answer is that yes, 7 of these dorsal glutamatergic neuron types really fall into like a one below the next, where you can see from the color that like they're getting more towards the dorsal. And then it sort of breaks down and bifurcates in the lamina 1 and 2. And so that's sort of an interesting collision between taxonomy, transcriptome, and spatial location. Another interesting thing we found is that there are these PIDX2 positive cholinergic interneurons, and these were identified in mouse. They haven't actually been seen in primates before. And these are the sort of sole source of cholinergic C boutons, which synapse onto spinal cord motor neurons. They're this cool, distinct population. And they form this very like small little column towards the medial part of the spinal cord. And you can see that we have like 1 or 2 cells per section. But because we have this in-depth transcriptomic information, we're able to actually pull those out and recognize that this column, which has been seen in mouse, indeed is also seen in primates. We also had another interesting finding where we found that across different species, we found that the dorsal horn in sort of the larger animals, so human is larger than macaque, which is larger than mouse, we see that in terms of how many dorsal horn neurons compared to ventral horn neurons these species have, it follows that progression. And interestingly, if you actually just look at the area of the dorsal horn compared to the ventral horn, it doesn't really reflect that. And so it seems like what's going on is that there's just an increased density in the dorsal horn of human and macaque. And yeah, I mean, you can speculate about why this is. It might be because we have larger bodies and so have larger sensory sort of fields that all this information needs to get channeled through the spinal cord. And so maybe it needs more efficiency for that, or we don't know. Functional studies may illuminate why this allometric trend exists in the future. We also looked at the sizes of motor neurons because classical anatomy has sort of long seen that mouse motor neurons are much smaller than human motor neurons. And so we looked in our macaque sections at these beautiful nu-N stains in pink, and we were able to quantify the sizes of these neurons, and we see that even within macaque, the motor neurons are different sizes across the segments, where cervical and lumbar are the largest, and then sacral is the smallest. We also can look at sort of across segments again, gene expression gradients, where we see that interestingly, the Hox genes are way more likely to be following sort of a roster-caudal gene expression gradient. And the reason that's interesting is because these are transcription factors, and you would expect them to be driving expression of other genes. But the sort of roster-caudal gradients seem to be much weaker in other genes than these transcription factors. And then doing gene set enrichment analysis, we see that the more anterior parts of the spinal cord are enriched for genes that are associated with more metabolism. So it's known to be more metabolically active. And then going more caudal, you see these anterior posterior pattern specification genes like the Hox genes. Yeah, comparing across species, we also see that this expected trend where human and macaque being primates are more similar. Macaque and then the primates versus the mouse are less similar in terms of their correlation. And we see a similar thing that we see in cortex, where oligodendrocyte progenitor cells are sort of more conserved, perhaps because they need to be progenitors and any change you make to them will propagate. But yeah, we see conservation of OPCs, which sort of stands out. Also looking at the sort of gene expression patterns, we use this measure from Jesse Gillis's lab, this ExpressiLog metric, which sort of is a 0 to 1 measure, where a 1 represents the pattern of expression is the same across all of our species, and a 0 is totally different. We find conserved families like chat, which are related to sort of the signaling and also like synaptic connectivity and stuff. We see divergent things like extracellular matrix, and very divergent. Really, the standout is link RNAs. And so using this ExpressiLog metric, looking at it compared to the specificity of genes, we're also able to find sort of transcription factors appear very significantly enriched amidst the most specific and most conserved. And this is sort of something you might expect, where these transcription factors are defining cell type identity. And you can see that in our little heat map, these they are specific, and yeah, they define the cell types and control gene expression to keep cell types in their identity. And I think for our last bit of transcriptome, we also have these cross-species analyses, and so we wanted to know whether or not different groups of genes, whether you can use these markers to map cell types across species. And we see that the conserved genes outperform other gene sets, which I think makes sense. Sort of by definition, if it's conserved, it should be having the same expression profile, which means a classifier can pick it up. But yeah, so we see that using these conserved gene sets that we find will help use our taxonomy across different species, because the conserved genes are they have the information necessary to do this cross-species mapping of cell types. All right, and then I'll hand it over to Nelson for our last piece on attack.
Cool. I'll try and keep this brief. So we defined our taxonomy based on RNA-seq, and that was a choice that we made. It's kind of the standard at the Allen Institute to go cluster RNA to define cell types, and then try and figure out how those types look in other modalities. So we have 10x multiome here, which is RNA and attack from the same cell. And it allowed us to, transfer our RNA types, T types, into the attack space and ask questions about conservation across species. So in attack-seq, really briefly, you can measure these peaks, which is where you see big pile-ups of reads from attack-seq, implying the DNA is open, and regulatory machinery can operate in that space to drive expression of genes or other machinery. So we looked at these peaks across species to try and figure out what parts of the DNA were uniquely open in human, what parts were conserved in their sequence, which were like you can lift it over essentially across species, and then also what regions had conserved open chromatin across cell types. So this is the most exciting place, I think, is to see where multiple species are using the same piece of DNA, peak region, to drive expression of genes. There we go. So when we take our cell types, these heat maps are showing on the columns the group level terms, and on the rows marker peaks for all those group level terms. And this is very black diagonal. So essentially this says that we can find really good marker peaks for all of our group terms, and these peaks are conserved across species and equally mark these group level resolutions. We can also look at peaks that are species specialized. So some peak regions are really good markers in human. They degrade into macaque, and then in mouse, those regions are really not useful to identify a cell type. So we find both deeply conserved elements of DNA as well as, rapidly changing parts through evolution. Um, Really briefly, we utilize what are called DNA sequence models. They train on DNA to predict the attack seek, and their goal is to understand the regulatory biology underlying peak regions. So transcription factors that drive enhancer function in cell types is the goal of these kinds of models. So from all these beautiful types we've defined, so I'm using the old names here. That's a mistake on my part. We can ask the question for, say, the GABA dorsal 10 group from Yadav et al. What motifs are inherent to peaks that mark that cell type? And, EBF2 are present in the DNA underlying marker peaks of GLUT dorsal 10. And you can kind of repeat this process for all these types we defined, understanding what, unique DNA elements are being utilized by various cell types. And that can help us understand regulatory biology, but also try to target different cell types across the brain and spinal cord. So with these models, we can also ask the question, how similar are species in their expression and attack space? So on the top here is the similarity of species pairs based on RNA-seq expression, so gene expression. And on the bottom is how similar the same pairs are based on their usage of TF motif grammars. And it's interesting to see that these motif grammars are deeply conserved across species, even more so than gene expression. So there are these core elements of DNA that all species use to define cell type, which is pretty exciting to see. The Institute, we do quite a bit of work trying to understand how we can take enhancers with adeno-associated viruses, AAVs, and use those to drive expression of a gene or a fluorophore in a specific cell type. We've done this a lot in the brain, trying to target like cortical types or basal ganglia types. There's publications out now on both of those kinds of tools. But the idea is you take an enhancer, you put it in the AAV, inject it into a mouse, and see what cells light up with a fluorescent fluorophore. In this case, this enhancer is lighting up cortex and basal ganglia, striatum at large. So we wanted to try this in spinal cord as well. So Tanya Daigle and Emily Kusick have some really great work selecting tools that are useful in targeting particular dorsal horn types. On the left, I'm showing one of the tools they selected that with our taxonomy, we're able to understand is targeting these upper layer GLUT 11-12 or TAC3 positive types. These are the itch neurons that, control the itch sensation. So, having a enhancer that can target these types allows for functional like exploration of how itch sensation can be modulated by affecting these cell types in various ways. I will leave it there with the epigenomics, but there's a lot more we did. Please read the paper and dig in more. Just another call out, we'd love to see um, everyone contribute to these efforts to build taxonomies. So these CAP pages are hopefully really useful, and we encourage your involvement. And that's all we had. Do you have some time for questions, Rachel?
Yes, we do. Questions that we have so far include, let's see. There were a couple questions that were asked during the talk that Matthew answered some of them. One of the questions was asking about different atlases for different species. I'm interested in a mouse atlas. We have many different kinds of mouse atlases at the Allen Institute. The original one is from about 20 years ago, which features in situ hybridization data and is most commonly referred to as our reference atlas. And then more recently, we also have single cell and spatial transcriptomic atlases for the whole mouse brain as well. So depending on what kind of atlas you're looking at, and the spatial transcriptomic data for the mouse atlas has also been registered to that common coordinate framework from the original histological reference atlas that we have. So we really do try to integrate all of our different atlases together across time. But yeah, really our most used mouse atlas is that original reference histological atlas, which you can find at brain-map.org. And again, now we have our single cell atlases and also some cross-species atlases for mouse as well, looking at basal ganglia and also motor cortex. So depending on what you mean by atlas, we have a lot of different resources there. Another question that was asked was, how do you enrich motor neurons in macaque, and why didn't you do it for humans? Motor neurons do not express NUN, do they? Thanks. And then Matthew responded, we use the gene BCL6 for FACS. Is there any other, you'd like to elaborate further on that, Matthew, or is that?
Yeah, so I mean, it's not necessarily, I think they express slow levels of nu-n. It's just that they're very difficult to capture. They tend to be fragile and rare. And so you need a marker like this to really capture large numbers of them. And you also need to use a whole large amount of spinal cord tissue, which I believe is why we didn't capture human enrichments. It was sort of a tissue availability thing. Yeah, I mean, we would love to also capture human motor neurons, but we also have collaborators who are working on that. And yeah.
Yeah, and it turns out that even with a good like marker gene like BCL6 to do fact sorting on the technology aspect, it has to really be optimized to capture motor neurons. So, we used a whole macaque cord essentially to enrich for motor neurons and ended up with a few hundred. So, these samples are precious, and until we get it right, I wouldn't be comfortable. using a human sample with such unoptimized results. So there's ethical considerations as well and why we don't use human cord for all these kinds of enrichments.
Okay, another question that we have is, how do you deal with species-specific gene expression? Do you leave only ortholog genes in each data set and then integrate?
Yes, we map one-to-one orthologs and then do the integration problem. Um, it's of course hard to compare genes that you don't have a known match with, but we did try that with there are tools called Saturn and other methods that try to use um, the coding DNA sequence of genes to try and match them up across species. So we did go down that route a little bit, but to do the bulk of this work, it was on one-to-one orthologs, matched up across species.
Yeah, there is a little secret, which is that SCVI can handle if you just fill missing genes from other species with zeros, and you give it the species as a covariate. It can integrate just fine, and you can keep all those genes in there. You just have to keep track of which ones got filled with zeros. But there may be species-specific gene information that might help with clustering or whatever. Um, but that's not what we did here.
Another question that we have is there any study that maps retro-labeled neurons from each segment of the mouse spinal cord and differences in their gene expression and electrophysiology?
I know there are retro labeling experiments that have been done in mouse.
Yeah, there are many older ones.
Yeah.
Actually, our collaborator, Alan Herman Poole from UT Southwestern, is doing this with spatial transcriptomics in a study which should be on bioRxiv in the next 2 to 3 weeks. So you can keep your eyes open for that, um, and then you get it with all of the transcriptomic detail, and so you can see who's projecting to like pons or red nucleus or cerebellum. Uh,
And we have collaborators internally that are doing the reverse, where you do retro labeling from cortex down into
mhm,
spinal cord to see where these like long range projecting neurons of like layer five are contacting in the spinal cord. So,
mhm.
and of course, e-physiology is a component of that as well. So, a lot of work going on in this space. It's really interesting questions to know how cell types work in conjunction to control like motor function.
Yeah, because regarding patch-seq data, I'm trying to think. I mean, I know we don't, I believe, have any published patch-seq spinal cord work. Is that, yeah, are either of you aware of, yeah, ongoing work with that at the Institute?
Not at the moment.
I don't think we're doing that
Yeah.
at the moment.
Okay. Another question that we have is, what is significant about including long non-coding RNA sequencing in this work? Why did you look into it? I'm not familiar with it, and I did not quite understand what that meant.
Oh, the link RNA point was um, just it popped up when we were looking at which genes are not conserved in terms of their expression profile across species. Um, and so, yeah, I think that's really the only point about it that we want to make, is that, I mean, and this is well known that link RNAs, I mean, for one, are not great cross-species markers, um, for the reason that they tend to turn over in evolution very quickly. And a link RNA that exists in human, um, a large fraction of them are not going to exist in a mouse as well. And mouse is going to have many link RNAs that don't exist in human or any other species, just because they're such a rapidly evolving in terms of link RNAs being born and link RNAs being destroyed in evolutionary time. That's all we wanted to say, I think, unless.
We have about another minute or so left and a few more questions, so I'll just try to get through these quickly. And while we do that, I'm also just going to do a very brief survey that'll pop up on your screen that'll just ask you about this webinar. It's anonymous, and this is just helpful feedback for us to give to the speakers and also for us to collect about how useful these webinars are. And also just future ideas. If there's something we haven't covered that you would like us to cover, there's a spot to fill that in. One more question is, can you talk about identifying dorsal horn species, conserved neuron subtype specific enhancers using a single species for each discovery effort rather than trying to do it across species? This is referring to what kind of conservation you found across species for the open chromatin peaks and dorsal horn neuron subtypes. Where was it high or not? Where was it highest? Does this include just human or macaque and mouse? Matthew and Nelson, can you all see on your screen this full question typed out since it's a little long?
Yes.
Okay.
So I think, in the RNA space, it was pretty clear that most of these dorsal horn populations were quite well conserved and distinct across species. I don't recall a case, Matthew, you might recall that there was ambiguity in distinction of dorsal types based on RNA alone. I think when we got like mid and ventral horn is where that got a bit more confusing. But thinking about the conservation and like the attack seek data, um, we didn't know we were going to see, I guess. And it seems like there are really uniquely open regions in these various dorsal types, and it makes sense because again, these are all processing different types of sensory information. Itch neurons, like, an example we had. There are many other types of neurons that require specialized function, so it's not totally surprising to see peaks that mark them and that are conserved in species that all have an itch sensation, right? So that'd be my 2 cents at least.
Okay, and then let's see. Is any of these data available on Nemo yet?
Very soon, if not already.
I believe they're not yet available on Nemo. They're in the processing pipeline
Yeah.
and have been for maybe a month and a half. But they will be available imminently.
Okay, and I did also post the link to the preprint that Matthew and Nelson were presenting today in the webinar chat. I also added a link to the cell annotation platform. And I'm also going to put in the chat the link to the original spinal cord atlas that was made from the Allen Institute. I don't remember which year, but it was a number of years ago. And again, that's in situ hybridization data, which I believe Nelson might also be answering in there potentially. Yes, we're a little over time.
Yeah.
So unless there's one more question, I also just want to make sure we're respecting everyone's time. And again, this will be recorded and we'll send out, again, those links are in the chat, so please use those. And you can also go to, I'll type it in the chat, but community.brain-map.org is another way to directly interact with the scientists at the Allen Institute. It's a community forum where you can post questions. And you can have your questions answered by the different scientists here. And thank you all for participating in the poll as well. All right, thank you, Matthew and Nelson, and thank you everyone for attending this webinar. I know we had to reschedule, so thank you for your patience with that. And I hope you all enjoyed learning about this new project. Thank you.
Thanks all.